if the land is sort of kind of, tired and overmanaged, you can introduce mycorrhizal inoculants to the soil to give it some kind of a starting point from which to increase the natural abundance of these fungi in the soil.
It's great to be back with another Environment Edge podcast, and you're very welcome to the show. There's so much to learn about the microbial communities and interactions in our soils, working hard to maintain soil health.
Dr. Olga Lastovetsky, Assistant Professor in UCD's School of Biology and Environmental Science, joins us to tell us all about our fungal and bacterial allies and how these relationships boost crop production.
I'm Cathal Summers.
And I'm Georgia Glenn.
Your Teagasc Sustainability Advisors. And we're back for another Environment Edge podcast, number 111, and here we go.
Olga, can you tell us a little bit about your work with AMF? Exactly what is it and what kind of work do you do in soil health?
AMF stands for arbuscular mycorrhizal fungi. So they are beneficial soil fungi which form associations with the roots of the majority of land plants, including all major crop species, actually. And so how they work is they grow into the plant root, and then they extend out into the surrounding soil environment using their kind of thin tube-like cells called hyphae. And they forage for essential mineral nutrients, bring them back to the plant, particularly phosphorus, but also nitrogen. And they exchange it. The plant gives them in exchange for these nutrients, photosynthesis-derived carbon. So they are nutritional symbiontic plants. They help plants grow. But they also can protect them from diseases and from abiotic stress, particularly drought. So they also help with water acquisition for the plant. So that's one group of fungi that I work on. And my particular interest is actually in a group of bacteria that live inside these AMF fungi. And so we call them endobacteria.
You just named the several things on soil functions that the fungi help with. Are we really heavily reliant on this fungi? The way you described it, it just seems that without the AMF, that we're in serious bother. That there's so many interactions and to do so much work for us. It's particularly in agriculture.
Yeah, that's true. So these fungi, in terms of kind of all plants on Earth, they're actually credited with helping plants colonize land like 470 million years ago, because those early plants, they didn't have true roots. They had these small kind of hair-like things called rhizoids that just helped anchor the plant in the soil and weren't particularly good at foraging for nutrients. So these AMF, these arbuscular mycorrhizal fungi, they were also there at the very beginning when plants were colonizing land, and they're known and they've been shown to be associated with this early plant and actually help them forage for those mineral nutrients that they couldn't do themselves because they didn't have true roots. So in that sense, they're, like I said, they're credited with the ability of plants to really become so successful on land at the very beginning. And then, of course, the plants diversified and grew bigger and taller and evolved their own roots, but they maintain these what are called mycorrhizal relationships. And in terms of agriculture, yes, there actually have been people have been very interested in arbuscular mycorrhizal fungi, specifically in agriculture, because of these nutritional benefits. And so there's rising interest in AMF as alternatives to these, non-renewable and energy intensive to produce mineral fertilizers. In terms of practical applications, though, it seems that the uptake of using them in agriculture is not sort of lagging behind our theoretical knowledge of the fact that they would be useful. But in terms of how to practically apply it in an agricultural scale, it's still a little bit unresolved.
Olga, are the endobacteria, are they harmful or can they be helpful?
Research on AMF is quite widespread. We know a lot about the biology of AMF fungi, how they interact with plants, the kind of environmental factors that affect this interaction, the molecular factors that affect them, the ecological. But Recently, and when I say recently, really we've known about these endobacteria since the 1970s, but they've been kind of overlooked. But the endobacteria that live inside these AMF, they literally live inside the cells of these fungi, the hyphae, and then the spores, which are the reproductive structures of these fungi. They're quite widespread. So some of the research that I've done during my PhD and during my postdoctoral work was to show that in nature, 70 to 80% of all AMF harbor these endobacteria. But what they actually do inside the fungus is in terms of experimental evidence is unknown. And so one of the major projects that my group is focused on at the moment is trying to understand what these endobacteria do in AMF, which represents kind of probably a super important part of their biology that we know nothing about.
How do you actually study it? What's the process?
Very, very good question. So I think one of the reasons that people still don't know what these endobacteria do. And so I'm not giving you all the story yet. There are two different types of endobacteria that are known. One of them is a lot less common in nature, in AMF in nature, and we actually do know what they do. Let's call them Endobacteria A. We know what Endobacteria A do, and they are beneficial. They actually promote this reproduction of the fungus and the growth of it when it germinates initially. The other endobacteria, the one that's called Endobacteria B, the one that I'm particularly interested in, we know nothing about. So one of the major reasons of why we don't know anything about this other endobacterium is because AMF are actually quite hard to study in the lab, because they are very hard to grow in the lab, because they are what's called obligate, the AMF themselves are what are called obligate biotrophs, meaning that they actually absolutely require their plant host in order to grow and reproduce. And so that means that if you're bringing these fungi in the lab to study the biology of the endobacteria that live inside of them, you have to be able to grow them. And that means to be able to grow them, you have to have the plant host. So there are these cool techniques that have been developed, not by us, but by other people that we're trying to use at the moment, is to basically establish this, what are called in vitro cultures in the lab, where you grow plant roots. And an association just with the plant roots in plates, you have the fungus. And then we're trying to study the bacteria that's inside the fungus that's inside the plant. So it's not entirely trivial, but when we get it to work, it'll be really cool.
So are you actually studying how the endobacteria change how the fungi interact with the plants?
Yes, so we're trying to understand everything because we don't know anything about how the endobacteria affects the AMF and how it might affect the interaction between the AMF and the plant host. Yes, we're trying to understand, trying to get kind of a basal level of knowledge about all of it. So the plan is to generate these AMF isolates that are identical in every way genetically, but they differ in the presence and absence of this endobacterium. And once we get them, we can then compare how these fungi grow with and without bacteria, how they interact with the plant host, how well they colonize the plant host, how they, interact at the molecular level, and then eventually if this presence of endobacterium inside the AMF affects plant host physiology, growth, reproduction, yield, biomass, all of these parameters where we would be measuring.
Olga, I suppose when we bring it back to the practical implications on farms for agriculture,
Mm-hmm.
you spoke at the start about how it's a kind of relationship between the plant and AMF and the bacteria altogether, and they exchange carbon for nutrients. It kind of work the other way as well, where it actually holds nutrients back if it doesn't get the supply of carbon at once and locks up our nutrients.
So there's been a lot of really cool research on that by a group in Amsterdam. They showed that, so a lot of research on AMF has been done from the point of view of the plant, because we're interested in the plants and, they have agricultural relevance. But this group, where they actually showed that the fungus is an equal partner in the symbiosis, in that, yes, in certain cases, if the plant isn't able to give it enough carbon, it will withhold the nutrients. And vice versa, the plant will not provide the fungus with a lot of carbon if it doesn't get enough nutrients in return.
Is something like clover on farms, is that an example of it?
Clover is a what's called a legume, and they can form symbiotic associations with a special group of nitrogen-fixing bacteria called rhizobia. So that's another example of how an interaction between plants and soil microbes can improve the nutritional status of the plant. And actually, clover is also mycorrhizal, so they can not only form relationships with these nitrogen-fixing rhizobia bacteria, they can also form relationships with mycorrhizal fungi.
Are some plants, Olga, better at forming genetic relationships or partnerships with the beneficial microbes in the like AMF?
Yes, and it's a very interesting also area of research. So the crops that we grow today, they have been bred in a very kind of a perfect environment for them. They have been provided with overabundance of nutrients and a perfect environment for growth. And in this environment, where there's overabundance of nutrients, the plant doesn't really need its mycorrhizal fungal partner, right? In that sense, the mycorrhizal fungal partner would be almost kind of a drain on its carbon source. So in that case, so there is what's called this mutualism parasitism continuum in any symbiotic interaction, that depending on the environmental conditions, if there is, say, an environmental condition where there is enough nutrients in the soil, the plant will not need the mycorrhizal partner as much. And in that sense, the mycorrhizal partner will just be a drain on carbon, and so will be less mutualistic and might even veer into the continuum of being parasitic. So in that sense, unfortunately, the crops that we routinely grow, they haven't been bred to be symbiotic with mycorrhizal fungi. So when you're trying to apply mycorrhizal fungi in an agricultural setting, there is a variation of responses from these crops. In some cases, they can benefit a lot from the mycorrhizal association, but in other cases, that's actually been shown that the mycorrhizal fungi can reduce the growth of these crops. And that's also a really cool area of research. And recently there was a study that came out of Switzerland, where they inoculated mycorrhizal fungi on 54 different fields in Switzerland, and they grew the same crop. They grew corn in every old field, and they did this study over 3 years, and they found that in some fields, the mycorrhizal fungi improved the biomass of corn by up to 40%, whereas in some other fields, it actually reduced it by as much as 12%. And so they were trying to figure out what is it that explained this variation of growth responses to mycorrhizal fungi. And they actually found that the biggest factor that explained this growth variation were soil microbes. So this is where we're kind of coming back to this tripartite interaction between plants, mycorrhizal fungi, and microbes that live in the soil.
So the microbes are the problem, Olga, are the benefit of the problem. But if you were the farmer and you wanted to grow more, what can we do then to make sure that we have the correct biology in the field, or to improve our soil health to get better crops off the back of it?
Yeah, of course. So that study concluded that knowing what microbes are in your field to begin with can predict the level of success the mycorrhizal fungi or mycorrhizal inoculations will have. And actually, in that specific case, the finding was that if there were already in that soil a lot of fungal taxa that are known to be pathogens, that predicted that meant that the mycorrhizal inoculations in that field would significantly improve crop growth. But if those particular fungal taxa that are not pathogens were not in the soil, then in that case, mycorrhizal fungi might not improve growth.
Just on that one, actually, because sometimes we think pathogens are bad, but it actually improved the growth.
It wasn't the pathogens that improved growth. I guess, sorry, I didn't explain this very well. The conclusion then was that the mycorrhizal fungal inoculations in those fields were they ended up protecting the crops from the fungal pathogens in the soil. So their benefit was due to as a result of protection from plant pathogens that were in the soil to begin with.
Excellent.
Olga, are some of the plants immune? Have they an immune system, which determines, say, whether they're going to allow the microbes to stay or not stay within the root zone?
Yes, plants have quite a sophisticated immunity. All plants have an immunity, and yes, plant immunity does determine what microbes it will allow to associate with. But and it has been shown that different plants in the rhizosphere or in the root zone will what's called kind of assemble a specific community of microbes, kind of like, humans, different humans, depending on their environment, they will have different gut microbiomes. It's sort of the same thing there. And I don't know if it's to do specifically with differences in immunity or differences in, production of specific root exudates that are particularly tasty to a particular group of microbes. There's a lot of factors there, and a lot of factors explain it. But yes, the plant host genetics do explain the kind of microbiome that they will assemble around themselves.
So there's better integration with some genotypes for AMS.
Yes.
Yeah.
Yes, so it is. Yes, so I think you're probably thinking, is it possible to genetically to breed or better to be more beneficial to derive more benefit from these microbes? And the answer
Yes.
is yes. Yes, and I don't. Yes, I agree. Yes.
It's quite complex, isn't it, Olga? And on the one hand, I suppose if we put too much chemical fertilizer out, it breaks the system a little bit, and it affects our soil health, and the system doesn't work as well. So, is there a difference, say, between, say, really intensively farmed grasslands versus maybe, or even croplands versus, more natural or semi-natural grasslands? Do you see a difference in the microbial communities that might affect how AMF works?
That's a very good question. And again, unfortunately, the answer is also quite complex. In more intensive management, depending on what kind of intensive management, but yes, more intensive management is associated with a reduction in the diversity of, specifically in this case, AMF or about mycorrhizal fungi. More natural ecosystems tend to have higher diversity of mycorrhizal fungi. So there are two sort of main ways that people can apply mycorrhizal fungi to improve plant growth, whether it's an agricultural setting or a natural setting. You can introduce land management practices that improve or facilitate both the natural abundance and the diversity of mycorrhizal fungi that are already there, because they really are everywhere. So these practices include, no reduction or abolishing of any fungicides, because fungicides, whereas they target plant pathogens, they'll also kill other beneficial fungi in the soil, including AMF. Reducing tilling, not leaving land fallow. Because all those practices have been shown to reduce the diversity and abundance of mycorrhizal fungi in the soil. And then if the land is sort of kind of tired and overmanaged, you can introduce mycorrhizal inoculants to the soil to boost, to give it some kind of a starting point from which to increase the natural abundance of these fungi in the soil.
Can you see any key research gaps within your area of work that might need to be addressed?
Yes, absolutely. There are definitely some. So, in terms of application of mycorrhizal fungi and mycorrhizal fungal inoculants in agriculture, there are definitely some key gaps. We know a lot of theoretical knowledge, and because these fungi have been shown theoretically to help, particularly in greenhouses and sometimes in the field, there are actually already a lot of products on the market that boast, mycorrhizal inoculants for crop growth. But the problem is that a lot of those those products are not tightly controlled, and so they tend to have very variable responses in the field. And so because the responses are variable, sometimes they work, sometimes they don't, sometimes they actually make things worse, the growers or the farmers don't tend to trust these products. And so the key research gap, I think, is for a structured collaboration between researchers and the industry which produce these inocula to get together and actually address the best way, the best mycorrhizal species, the best inoculum potential, address some of these key questions that will then improve the efficacy of these inocula in the field and improve, which would improve farmers' trust in these products if they are actually helpful to plant growth.
Just one question, I know the inoculants can be a minefield because there's so many of them out there maybe and different things, but can we go something more simplistic and maybe try and improve organic matter on land? Would that help towards it as well?
I suppose in terms of mycorrhizal fungi, it's hard to predict because, plants get their organic matter, they make their own organic matter by photosynthesis, and then mycorrhizal fungi don't actually forage for organic matter. Really, they, especially for carbon, they get it from the plant. But in terms of improving soil, the bacteria and maybe other fungi in the soil, yes, I think it's definitely, I think as long as the soil health is improved, all the other pieces might fall into place. I know that's probably not very scientific. It might be more poetic, but I think once the ecosystem is healthy, it is healthy, and improving soil organic matter might be one of those.
So looking forward then, Olga, to the near future, I think by what you're after telling us during the pod, are there any biotechnological applications that you envisage arising from the kind of research that you're doing for, say, example, designing, say, smart biofertilizers like nitrogen fixing, phosphorus, bacteria and fungi?
Yeah. So in terms of my own research, how it has applications. So if we're researching this endobacterium that's very common in mycorrhizal fungi, and mycorrhizal fungi are being developed into these, biostimulants. I mean, we used to call them biofertilizers, but they're now under the umbrella of biostimulants, as the EU directive defined it. In terms of my own research, once we know what these endobacteria do, if they are helpful to the fungus, yes, we can then we can design these biostimulants, these mycorrhizal biostimulants, to have these endobacteria. If we find that they are detrimental to the fungus, which I really don't think so because they're so common, then we can design these mycorrhizal biostimulants to not have them. So this is kind of a very simplistic way, but you're right. Yes, knowing the biology of the interaction between mycorrhizal fungi, plants, and microbes in the soil and in the plant roots, we can design, smart, or what are they called, synthetic communities. So, if you inoculate a field, not with just a single species of mycorrhizal fungi, but maybe it's a mix of different species of mycorrhizal fungi and different bacteria that are known to help mycorrhizal fungi in plant colonization. Yeah, going forward, I definitely think there is potential for that.
Olga, we've learned an awful lot from you in the last 20 minutes, and I think there's so much more to learn as well. So I think we're going to have to get you on again another day to dig in, pardon the pun, dig in deeper into more AMF and fungi and bacteria, and there's so much going on there, and it's clear to see there's still a lot to learn even on the research side as well. It's a vast world, but just like to thank you for joining us on the show today and keep up the good work.
Thank you so much for having me.
So there you have it. Tune in next time to hear more interesting chats and discussion.
Myself and Deirdre will be back in 2 weeks' time, but do give us a review on Apple and Spotify or wherever you get your podcasts. Thanks for listening to the Environment Edge podcast.